Hello students, and welcome to our next lesson in the Central Dogma unit. Today we're going to be talking about DNA replication. So to kick off this lesson, let's pretend it's the middle of summer again. You went to the beach and you soaked up lots of sun, but you forgot to wear your sunscreen. So you got a really bad sunburn and your skin started peeling over the next few days. However, you noticed that underneath that layer of damaged skin, there was already a new layer of skin. What caused the skin to peel, and how did our body create the new layer of skin? Well, UV rays from sunlight actually damage the DNA in skin cells, and too much damage signals these cells to self-destruct. But luckily, we can make new cells by the process of mitosis. So we're able to build a new layer of skin cells that will replace the old ones. Now, how does this relate to DNA replication? Well, in order to perform mitosis and make those new cells, we first have to replicate our DNA. DNA replication is absolutely necessary before mitosis, so without DNA replication, we can't do mitosis, and without mitosis, we can't heal our sunburn. And this is just one little example of the importance of DNA replication. So what exactly is DNA replication? Well, DNA replication is the process of copying DNA before cell division. So remember that cell division in prokaryotes is what we call binary fission. And our first step in this case is DNA replication. Now, cell division in eukaryotes can either be mitosis or meiosis. So we see an image here of the cell cycle. And I want to point out that during this S phase of interphase, this S phase is what we call our synthesis phase. And this is where DNA replication occurs. And we notice that this DNA replication has to occur before we can reach the mitosis phase. So in both of these cases, whether that be prokaryotes or eukaryotes, DNA replication must occur before cell division. Because in cell division, we're splitting up the DNA equally between cells, so we have to have twice as much DNA to start with. And that's what DNA replication does. It makes two copies of DNA so they can be separated into different cells. Here we can see two diagrams showing the overall process of DNA replication in prokaryotes and eukaryotes. So I want to point out a few things. Remember that prokaryotes have circular DNA. So they have circular chromosomes, whereas eukaryotes have linear DNA. Now, since prokaryotes have circular DNA, when they go to start the process of DNA replication, they're only going to have one origin of replication. And an origin of replication is just where DNA replication starts. However, in eukaryotes, we actually have a bunch of DNA. So we're going to have multiple origins of replication in order to speed this process up. So we end up with a bunch of replication forks here because we have many origins of replication in order to speed this process up. But the end result of DNA replication in both prokaryotes and eukaryotes is the same. So in both cases, DNA replication makes two daughter DNA molecules. So we start with one DNA molecule and we're able to copy it and make two. Now, DNA replication follows what we call semi-conservative replication. And this means that in our final DNA molecules, one of the strands will be from the original DNA molecule, which is shown in pink here, and the other strand is a newly created strand. So if we look over here, we can see that we start out with our original or our parent DNA molecule. All right, and as we separate this DNA molecule and start to add our new nucleotides, these strands shown in pink here are going to be our template strands. And what we're forming in blue is our new strands. And overall, we end up with 2 identical DNA molecules. So remember when we talked about our base pairing, our A is always paired with T and our G is always paired with C. And when we look at these DNA molecules that we formed, we can see that they are exactly like our original DNA. So in our DNA replication, we're taking one DNA molecule and we're making 2 identical DNA molecules. And in each strand here, we essentially have half old DNA because it's from our parent strand and 1/2 new DNA, and we call this semi-conservative replication. Another important thing to note is that DNA replication always occurs in the 5' to 3' direction. So remember, we called our 5' end like the starting point and the 3' end the ending point, and DNA replication will always occur from 5' to 3'. So when we look at this leading strand here, this is the strand that's going to be made toward the replication fork. So right here, we call this a replication fork. And our leading strand is going to be made towards the replication fork. We can see it's being made 5' to 3'. And this is going to be continuous DNA synthesis, meaning there's no gaps because we're moving toward our replication fork. On the other hand, when we look at this lagging strand down here, we can see our DNA synthesis is still from 5' to 3', but in this case, it is discontinuous because we're forming only these short fragments of DNA with gaps between them. And we call these small pieces here Okazaki fragments. And we're going to talk about those in just a second. But overall, I want you to note that whether it's the leading strand or the lagging strand, either way, DNA replication is occurring in the 5' to 3' direction. Here we see an overall diagram of DNA replication, and we're essentially going to have 3 main steps. So before we can start replicating our DNA, we need to unwind those strands. We need to unzip those hydrogen bonds between our bases. And to do so, we're going to use the enzyme called helicase. So this ACE here indicates that this is an enzyme. And our first step, helicase is going to come in and it's going to unwind our DNA and separate the hydrogen bonds between bases. And now that we have 2 template strands formed, our DNA polymerase, which we can see here and here, our DNA polymerase is going to come in and add nucleotides to form our new strands of DNA. So if this was in the dark blue up here, our parent strand, let's say this little sequence here was ATGC, our DNA polymerase is going to come in and add complementary bases. So remember, A pairs with T, T pairs with A, G pairs with C, C with G, and so on. So our DNA polymerase is coming in and adding those nucleotides on both the leading strand and on the lagging strand. Now when we look at our leading strand here, our synthesis is from 5' to 3', and this is continuous. So there's no gaps in our DNA. But on our lagging strand down here, we can see how we're only making short fragments of DNA. And again, we call those small pieces Okazaki fragments. So on our lagging strand, our DNA synthesis is discontinuous. So we end up with a bunch of little pieces of DNA, and we want to be able to put them together. And the enzyme that puts them together is DNA ligase. So we can think of DNA ligase like a glue. Our DNA ligase will come in and seal the gaps in that lagging strand. And this will help us make a continuous strand of DNA. And as we go through this process, our end result is going to be taking that original DNA molecule and making 2 identical copies.